This presentation gives a brief introduction to some of the biological and social factors that affect our awareness of gender. The presentation questions the traditional distinction that sex is determined biologically and gender is determined socially. It also questions the binary distinction between males and females. The current model of thinking about gender is the separate idea of sex, a binary distinction between being male or female, from that of gender, which is a more sophisticated concept. The sex of a child is thought to be determined biologically by the appearance of sex chromosomes XX or XY in the nuclei of the cells, or the appearance of the external genitalia which are inspected at birth. This is the sex attributed to a child by the midwife or the hospital staff or by the parents. Gender, on the other hand, is regarded as being socially constructed and has been built into the ideas of gender identity, whether you regard yourself as being masculine or feminine, as well as the role of that gender plays in society. Because it is a social construct, then gender is influenced by family and friendship groups, as well as the wider media. The basis of the binary distinction of sex into male or female is traditionally associated with the presence of a Y chromosome in males. There is one gene in particular called SRY, which becomes active at around about 6 weeks of embryonic development. The activity of this gene leads to a rise in hormones associated with masculinity, the so-called androgen hormones, and testosterone in particular. The rise in testosterone activates cells which develop into testes. This changes the development pathway of the embryo from one that is essentially female, resulting in ovaries, uterus, vagina and vulva, into one that is essentially male, involving testes and a penis. There is then a close link between the biological sex, the presence or absence of the Y chromosome, and the sex attributed at birth. The observation that many people are uncomfortable with the sex they were assigned at birth when developing their own gender identity suggests that the story might be more complex than might first appear. Some people have a version of SRY gene which does not result in the production of androgens, or their cells do not respond to the androgen hormones produced. In such cases, their development continues along a female path, even though they have Y chromosomes in their cells. This is referred to as complete androgen insensitivity syndrome, or CAIS. People with this condition have a biological sex that is male, but an attributed sex that is female. Usually such people are happy to develop a feminine gender identity and they live their lives as women. They are, however, unable to have children because the ovaries or the uterus may not form properly. However, it is also clear that intermediate conditions also exist, so-called partial androgen insensitivity syndrome, or PAIS. Here the SRY gene does stimulate the production of some testosterone, but the amounts produced are suboptimal for the regular development of the testes. In this particular condition there is considerable variation in the appearance of the external genitalia, which may be intermediate between male and female types. Such people are often referred to as intersex. Because gender is socially constructed, it is traditionally associated with non-heritable family shared environments. and also with non-shared environments. Here is an interactionist model of the mind. The genome interacts with body cells, and the mind emerges as a property of the ways that the cells of the human body interact together. Self-identity is one of the features of the mind that emerges from these interactions. Gender identity is a subset of self-identity. Therefore, one's gender identity emerges from an interaction of the genome with the body cells, and of the body cells with each other, as well as the effects of shared and non-shared environments. People whose gender identity matches their biological sex or their attributed sex will align somewhere on the gender identity scale, so-called cisgender. For people whose gender identity does not match their biological or attributed sex, they will align in different places on the gender identity scale, so-called transgender. The potential conflict may be resolved at a point at which the person is comfortable to self-identify. Human Diversity is a complex but important book which looks at the biology of gender, race and class. Charles Murray became a very controversial writer in the 1990s when he wrote a book called The Bell Curve. Murray takes a hereditarian position and argues that genetic differences can explain the variation in gender in human society. He also argues that there is a biological basis for traditional gender roles. The rest of this presentation looks at some of his arguments in detail. Murray's book is built around a series of 10 propositions. This is the first proposition. His argument is that differences between the sexes for personality traits have a biological basis, which ultimately depends upon the genome. He argues that this is the same across all cultures, and the differences are even more pronounced in those societies which have created much more gender egalitarian social structures. At the heart of his thesis is the idea that men are interested in systems and mechanisms, whilst women are interested in people and feelings. This is an idea that comes from the behaviourist Edward Thorndike. It accords with something the psychologist Sacha Baron Cohen has proposed, that on average men tend to be systemizers, while women tend to be empathizers. The emphasis on average is important because there are considerable variations within each group, and these effects exist within the spectrum of continuous variation for these characteristics. One way to think about variation in complex characteristics is to think about effect sizes. The graph above is taken from Murray's book. It shows the distribution in height for male and female populations in America. It shows that the mode or the mean value for height differences in males and females is different, but there is considerable range in both populations. There are many women who are taller than shorter men. However, the tallest people are men and the shortest people are women. The variation within each population can be expressed as a mean and standard deviation from the mean. The effect size D is a way of comparing the magnitude of differences between the two populations. It can be calculated by subtracting the male mean from the female mean and dividing this by the pooled standard deviation. This is the standard deviation of the combined population. The importance of a factor depends upon the magnitude of its effect size, and values range from 0.2 to 0.5 have small effects. Those with effect sizes between 0.5 and 0.8 are medium effects. Anything with an effect size larger than 0.8 has a large effect. The sign in front of an effect size statistic is usually ignored. Here are some effect sizes for a study carried out in 2005, which looks at 50 populations across the world. You can see that for characteristics like shows warmth towards each other, small to medium effect sizes are seen across all of the different nationalities, with females having the higher scores. The same is true of an appreciation of art and beauty. Notice that this is not saying that all women show warmth towards each other and appreciate art and beauty, whilst men do not. It is saying that in populations, a slightly higher proportion of women show warmth towards each other than men, and also a slightly higher proportion of women appreciate art and beauty compared with men. However, the effect sizes are medium in magnitude, and therefore the variation within the groups is highly significant. The characteristics on altruistic concerns and sympathises with people show similar trends. In all cases, the females have higher means than the males, and the effect sizes across the different cultures are very similar. This suggests that these characteristics do not have a cultural basis, although they may be influenced by culture. It is indirect evidence for some kind of common biological effects. Murray's second proposition is that females have advantages in verbal ability and social cognition, whilst males have advantages in visuospatial abilities and extremes of mathematical behaviour. Pause this film for a moment and read the list of characteristics on the right-hand side, each of which is supported by a medium effect size. Murray's book continues with a list of differences. Where available, the effect sizes are shown. Again, pause the video until you're ready to move on. One interesting feature is that at school there are no differences in mathematical abilities between males and females. However, males predominate in higher level maths environments, such as undergraduate and postgraduate courses. This suggests that social influences might be putting women off studying mathematics at higher level, rather than a lack of ability. This is supported by the finding that there are no apparent differences between males and females for general intelligence, little g. Murray's third proposition is that, on average across the world, women are more attracted to vocations which are centred on people, and men are more attracted to vocations centred on things. What is clear is that social changes in the last century have allowed women to have greater access and control over their own lives, including their reproductive fertility. And this is reflected in the significant increase in the number of women who are obtaining professional degrees since 1960s onwards. Curiously, the figures for males have not followed a similar trend. This is a survey based on the work of Holland and Prediger, and it's a survey of nearly 250,000 men and women across the world. Their participants were assessed upon the grid with two dimensions, people and things. On average, women's vocational interests tilted towards occupations involving work with or understanding of other people. Men's vocational interest tilted towards working with things. The biggest shifts involved the realistic orientation, a male preference, with an effect size of 0.84, and also the social orientation, a female preference, with an effect size of 0.68. When the data were analysed along Prediger's 2 dimensions using his indexes, a striking contrast emerged. On the data ideas dimension, there was virtually no sex difference. On the people things dimension, the effect size was plus 0.093, meaning that women were on the people end and men were on the things end of the dimension, and this is a large effect size by any standard. Murray's final proposition is that there are structural differences between the brain that can affect the way that men and women think, which can explain the people-things axis. A new field of neuroscience called the connectome maps how brain cells, neurons, connect with each other. In general, male brain cells network most strongly within modules within the 2 hemispheres. In contrast, female brains connect most strongly with neurons between the hemispheres. Greater connectivity within hemispheres, combined with greater activity across the 2 hemispheres of the cerebellum, was found in males, leading to an increased competence on a spatial task in which they excelled. For females, greater connectivity between hemispheres would facilitate the integration of the analytical and sequential reasoning modes of the left hemisphere with the spatial intuitive processing of information of the right hemisphere. And this is consistent with competence on a language test in which females excelled. Research with mice suggests that exposing female fetuses to androgen hormones stimulates masculine characteristics in the developing female mice. There is no evidence of a reciprocal effect. Developing male mice are not feminized by the presence of estrogen hormones. Does this effect also apply to humans? Mothers do naturally produce androgen hormones during pregnancy, although the amount that they produce varies between mothers. Baron Cohen has found an inverse correlation between eye contact and vocabulary and the level of fetal testosterone. The higher the levels of fetal testosterone, the lower the levels of eye contact and reduced vocabulary in toddlers at age 12 and 24 months of age. There seems to be an interaction between biological mechanisms and social learning. Richard Udry measured the natural variation in androgen levels in mothers when they were pregnant. And then he also measured the participants along 4 masculine and femininity scales. Childhood gender socialization, the ability to learn gender characteristics through social learning, seem to be independent of the gendered behaviour of the mother with high levels of androgens. Social learning of gendered behaviour was unsuccessful if the mothers had got higher level of androgens during pregnancy. Alternatively, if the levels of fetal androgens were low, then gender socialization was more successful. Udry's argument is that the predisposition to be feminine is triggered by lower levels of fetal androgens, and this can be further enhanced by social learning within the family. The evidence in this presentation emphasises that whilst there are differences between males and females, the variation within each group is large. The data fits with the model that our sense of gender identity and our gender roles emerge through the interactions of our genomes with the effects on our bodies and also the wider environment. The wider environment includes the effects of our parents' genomes, especially of mothers during fetal development, and the maternal physiology. All of this variation is normal, to be accepted, cherished, and celebrated.